Right now, as you listen to this, your body is engaged in, well, a microscopic game of traffic control.
Oh, absolutely. It's life or death, really.
Yeah, millions of your cells are dividing, migrating, multiplying, dying all perfectly on cue. But today, we're doing a deep dive into what happens when just one of those cells decides to, cut the brakes.
Right, when it just floors the gas and builds its own rogue highway system.
Exactly. We're looking at a stack of medical presentations today to demystify the biology of tumors. We're unpacking the exact mechanisms of how a normal cell rewrites its genetic rulebook, and we'll also look at the clinical data on cancer's footprint across human populations.
It's a heavy topic, but an incredibly fascinating one.
It is. So, okay, let's unpack this. Because to make sense of something so complex, I feel like it really helps to think of those cells like cars navigating a perfectly synchronized city grid.
Right. Yeah, I like that analogy. Because, to truly grasp the destructive nature of cancer, we first have to just marvel at the absolute precision of that normal city grid.
Right, the baseline has to be established.
Exactly. Normal cellular programming is just astonishing. Our cells operate with these intricate checkpoints and, strictly regulated life cycles and built-in expiration dates.
So it's not like a foreign invasion?
No, not at all. When we talk about neoplasia, which just means new growth, we aren't talking about an alien invader. It is a corruption of your own incredible biology.
Wow, yeah.
Your own cellular machinery is essentially hijacked and turned against you.
Which is terrifying, but before we get into the truly rogue elements, the malignant cancers, we need to establish what a growth actually is, right?
Right, because not every lump is a villain.
Exactly. Because a traffic jam on our cellular highway isn't always a malicious driver. Sometimes it's just like a misplaced road. In pathology, not every lump is actually a tumor.
Yeah, take chorostomas, for example.
Right, let's talk about those.
The choristoma is fundamentally normal, perfectly healthy tissue, but it simply got displaced to the wrong organ during embryonic development.
By endometriosis, right?
Yes, exactly. Endometriosis is a classic example. You have normal uterine lining growing outside the uterus. So it acts like a mass, and it can certainly cause complications, but genetically, it's just lost tissue.
It's just confused.
Basically, yeah. And then you contrast that with something like a hamartoma.
Which is the right tissue in the right place, but proliferating into this disorganized mass.
Right. Imagine a construction crew building a house with all the correct bricks and timber, but they entirely ignore the architectural blueprint.
Just a chaotic pile of building material.
Exactly. You end up with a mound. Neither the chordostoma nor the hamartoma are true tumors. To observe a true benign tumor, we'd look at something like a lipoma.
Okay, the textbook benign growth.
Right. It's an encapsulated, mobile, painless mass of mature fat cells.
And if you put it under a microscope, from the sources we read, the cells are almost indistinguishable from normal body fat, right?
Completely. Just bright yellow adipose tissue separated by fine fibrous bands.
But I want to be clear here, because just because a tumor is benign does not automatically mean it's harmless. Location really dictates the danger here.
Oh, absolutely. A lipoma on a patient's arm might just be, a minor cosmetic annoyance.
Right.
But if that identical benign mass grows inside the major airways, it physically obstructs airflow. It causes severe respiratory distress.
Wow, just because of where it is.
Exactly.
Yeah.
Or an esophageal lipoma. The patient can present with severe swallowing difficulties, regurgitation, vomiting. They can even grow within the heart, projecting directly into the cardiac chamber and obstructing blood flow.
That's wild. So benign simply classifies its biological behavior, meaning it doesn't invade or spread, but it makes no promises about its physical impact on you.
Exactly. It's a classification, not a guarantee of safety.
But wait, if a lipoma is made of perfectly normal, mature fat cells, and it's not cancer, why does it keep growing at all? Like, why doesn't it just hit a certain size and stop, like a normal organ does?
That's a great question. Benign tumors do still suffer from a localized overgrowth. They have altered growth signals, so they're dividing more than they should be.
Okay.
But, this is key, they maintain a crucial biological boundary, which is encapsulation.
They stay in their lane.
Yes, they don't invade the surrounding architecture, and they completely lack the genetic tools to metastasize to other organs.
So understanding that physical encapsulation really sets the stage for grasping the sheer catastrophe of what happens when a cell actively develops the tools to, tear those boundaries down.
Precisely. That's the leap from benign to malignant.
Which brings us to the actual journey of a rogue cell. Because a normal cell doesn't just wake up one morning completely malignant and ready to tear through tissue.
It's a progressive, step-by-step unraveling.
Right. And the earliest stage of this unraveling is called hyperplasia.
Right, which simply means an increase in cell numbers.
Yeah.
The tissue is dividing more frequently, but the cellular structure, the histology, remains completely normal.
And crucially, hyperplasia is reversible, right?
Yes. If the environmental stimulus or stressor causing the rapid growth is removed, the tissue can revert right back to its normal state.
Okay, so then the progression moves into dysplasia. To use our crowd analogy from earlier, dysplasia is like a peaceful gathering getting a bit rowdy and disorganized.
I like that. Yeah, the cells start losing their proper orientation. They become enlarged.
But you can still calm the crowd down and send them home. Dysplasia is still reversible.
It is. The true tipping point, the absolute point of no return, is a stage called carcinoma in situ.
Carcinoma in situ, which translates literally to cancer in its place.
Right. At this stage, the condition is non-reversible. Genetically and structurally, these are now fully malignant cells.
Okay, so what does that look like?
You see massive morphological changes. The nucleus to cytoplasm ratio jumps significantly because the nucleus becomes enlarged and hyperchromatic.
Hyperchromatic meaning like darker under a microscope.
Exactly. It's darker because it's just churning out mutated DNA. However, structurally, the basement membrane remains intact.
And the basement membrane is essentially the cellular floor that physically separates the surface tissue from the deeper systemic layers of the body.
Right, it's the barrier.
So the rowdy crowd has turned into a full-blown riot, and the doors to the venue are locked. The situation is irreversible, but the chaos is contained entirely to that one room.
That is the perfect way to visualize it.
So the final fatal step into invasive carcinoma is when those rioters start manufacturing tools to literally break down the walls.
Yes. And the mechanical precision of how a cancer cell achieves invasion is diabolically clever. It requires this three-part coordinated attack.
Okay, what's part one?
First, the cell decreases its production of a protein called cadherin.
Cadherin?
Yeah, cadherin acts as the intercellular glue holding neighboring cells together. So by dropping cadherin levels, the malignant cell physically detaches from the tumor mass.
Oh, wow. It unglues itself.
Exactly.
Oh.
Second, it upregulates surface receptors called integrins, allowing it to anchor directly to the extracellular matrix below it.
Okay, so it drops the glue, grabs the floor.
Right. And third, and this is the definitive weapon, it secretes specialized enzymes called matrix metalloproteinases.
Matrix metalloproteinases.
Yeah. These enzymes function as chemical scissors. They physically cleave the peptide bonds of the basement membrane, literally chewing a hole straight through the cellular floor.
It manufactures its own chemical scissors to cut through the floor? That is insane.
It is. But the body isn't just a passive victim here.
What? There's a response.
When a tumor invades the deeper tissue, it triggers a massive host response called dysplasia.
Dysplasia, meaning what exactly?
The surrounding healthy tissue desperately tries to build a thick wall of dense, fibrous scar tissue to trap the invading tumor. It's a microscopic trench war.
A war that the tumor is genetically equipped to fight. Because to manufacture those chemical scissors and drop the cadherin glue, that requires a fundamental rewriting of the cell's genetic code.
Absolutely. And almost all tumors are monoclonal, meaning this staggering chain of events originates from one single individual cell.
Just one cell that accumulates enough genetic damage to bypass all the body's fail-safes.
Yes.
So going back to our car analogy, the genetic damage specifically targets the gas pedals and the brakes.
Right.
The gas pedals are your oncogenes. Normally, your body relies on proto-oncogenes, which are these essential, healthy genes that promote regulated cell division.
But a mutation flips them into oncogenes, effectively permanently flooring the gas.
And the most terrifying aspect is that oncogene mutations are dominant. It only takes one hit.
Exactly. You have two inherited alleles for each gene. If just one of them mutates into an oncogene, the gas pedal gets stuck.
We see this with the SMIC gene in Burkitt's lymphoma, or the HER2 gene in breast cancer.
Yeah, and another massive player is the RAS gene.
Right, RAS.
Which is found mutated in roughly 30% of all human cancers. It drives incredibly aggressive colon, lung, and pancreatic tumors.
But a stuck gas pedal alone isn't necessarily fatal if the car's brakes still function.
That's right. And that brings us to the tumor suppressor genes.
The brakes.
Yes. They act as the cellular brakes to halt runaway growth. But unlike oncogenes, tumor suppressor genes are recessive. They require a two-hit model.
Okay, so both alleles, the copy inherited from your mother and the copy from your father, must be mutated or deleted to entirely lose that braking function.
Precisely. You have to lose both for the brakes to fail.
And the absolute VIPs of the brake department are the retinoblastoma protein, or RB, and the legendary P53 gene.
Oh, P53. The medical literature often refers to P53 as the guardian of the genome.
That's quite a title.
It earns it. It is the single most commonly mutated gene across all human cancers, implicated in over 50% of them.
Wow, over half.
Yeah. Normally, P53 acts as a strict toll booth operator in the cell cycle. If it detects damaged DNA, it halts cellular division entirely.
Just stops everything.
Yep, giving the cell time to repair the errors. And if the DNA damage is beyond repair, P53 ruthlessly triggers apoptosis-programmed cell death.
It basically commands the damaged cell to destroy itself to protect the host.
Exactly. So when P53 is knocked out, cells with severely mangled, heavily mutated DNA are just granted a free pass to replicate infinitely.
Which provides the exact mechanism behind Li-Fraumeni syndrome.
Yes, a very tragic condition.
So this is a rare inherited genetic disorder where a patient is born with a mutation in just one of their P53 alleles. From day one, they only have one working copy of the guardian of the genome.
Right. And clinically, this results in a staggering 25-fold increased risk of developing a myriad of cancers: breast, brain, leukemia, bone sarcomas, often before the age of 50.
But wait, I have a question here. If tumor suppressors are recessive and require 2 hits to break, these patients still have one perfectly functional copy of P53, right? They still have a backup break line.
They do.
So why is inheriting just one broken copy so catastrophically dangerous?
It really comes down to statistics
Huh.
and the concept of cumulative damage.
Okay, how so?
Well, having trillions of cells constantly dividing over decades presents trillions of opportunities for a copying error. A stray UV ray, a chemical exposure, or a simple biological misfire can randomly knock out that remaining good allele.
Oh, so they're just starting halfway to the edge of the cliff.
Exactly. Furthermore, DNA repair genes operate essentially as the body's spell checker. They're continuously scanning billions of base pairs for typos.
Right.
When a patient starts with a has-broken spell checker, the statistical odds of catching the next typo plummet. Uncaught mutations breed more mutations.
It's a snowball effect.
Exactly, accelerating the breakdown of other regulatory genes in a cascading failure.
So the cell has floored the gas, severed the brake lines, and bypassed the spell checker. It's replicating out of control.
Yes.
But biology imposes strict logistical limits. Normal cells have built-in expiration dates, and tissue requires a blood supply. A tumor cannot grow to the size of a golf ball simply by dividing. It has to overcome physical starvation and cellular aging.
Right, it runs into physical walls.
And cancer relies on 2 distinct survival tactics to bypass these rules, which our sources refer to as immortality and vampirism.
I love those terms. So, biological immortality is achieved by manipulating telomeres.
Telomeres.
Yeah, these are repeating sequences of DNA that cap the extreme ends of our chromosomes, protecting the vital genetic information inside.
Think of telomeres exactly like the little plastic aglets at the ends of your shoelaces, right?
Yes, perfect analogy.
Every time a normal human cell divides, that plastic aglet gets snipped a microscopic bit shorter. And after about 50 or 60 divisions, the aglet is completely gone.
Right, and the chromosomal shoelace begins to fray.
Exactly. The DNA becomes unstable and the cell undergoes senescence and dies. It is an inescapable biological clock.
But to become immortal, the cancer cell has to figure out a way to manufacture endless plastic to keep the shoelace permanently intact.
And the mechanism it uses to manufacture that plastic is an enzyme called telomerase.
Right. Healthy, normal, adult somatic cells do not actively express telomerase.
They just don't have it turned on.
Exactly.
Yeah.
But over 90% of human cancers undergo a mutation that turns the telomerase gene back on.
90%.
Yeah. The tumor continuously rebuilds those protective chromosomal caps after every single division. The biological clock is destroyed.
Granting the cancer limitless replicative potential, immortality.
Immortality. But an immortal cell still requires oxygen and nutrients.
Right, which leads to the vampirism. A tumor cell must be within 1 to 2 millimeters of a functioning blood vessel to survive. If a tumor expands beyond a few millimeters, the cells trapped in the dense, crowded center are too far from the bloodstream.
They will literally suffocate, starve, and die.
So to survive its own explosive growth, the tumor has to feed.
And the clinical term for this is angiogenesis, which is the physiological process through which new blood vessels form from pre-existing vessels.
So to breach that 2 millimeter limit, tumors secrete potent signaling molecules, right?
Yes, primarily one known as VEGF, or vascular endothelial growth factor. Yeah. VEGF acts as a chemical distress beacon. It diffuses into the surrounding healthy tissue, co-opts nearby normal blood vessels, and forces them to sprout new capillaries that aggressively grow directly into the tumor mass.
Oh, wow. So the tumor effectively hijacks the host's vascular infrastructure to feed itself.
Exactly. It forces the body to build in a private highway.
But the host body fights back here, too, right? The body naturally secretes anti-angiogenic factors, most notably a protein called endostatin.
Right. Endostatin circulates and attempts to neutralize VEGF. It inhibits those rogue blood vessels from forming.
So deep inside the tissue, microscopic tumors are constantly locked in this brutal, invisible tug of war. The tumor dumps VEGF to build supply lines, and the body dumps endostatin to starve it out.
And tumor's ability to grow is strictly dictated by the balance of that single chemical equation.
That is unbelievable. So if the human body possesses all these sophisticated defenses, spell checkers, p53 guardians, endostatin blockades, we have to ask what initially pushes the system over the edge? What causes that very first critical mutation?
Right. We have to look at carcinogenesis, which is fundamentally a two-step process. It requires both initiation and promotion.
Okay, initiation and promotion. An initiator is what we typically think of as a true carcinogen, right?
Yes. It is a chemical, physical, or biological agent that directly damages the DNA architecture, causing a permanent mutation in the cell.
Like breaking the code.
Exactly. A cell that has undergone initiation is permanently altered. However, that mutated cell might sit dormant in the tissue for years, doing absolutely nothing, because it hasn't been given the signal to divide.
Ah, and that signal comes from a promoter.
Exactly.
But wait, if a promoter doesn't actually damage or mutate the DNA itself, why is it considered such a critical threat? Isn't the damaged DNA the entire problem?
The damaged DNA is the blueprint for the problem, but a blueprint sitting in a drawer builds nothing.
Okay, that makes sense.
A promoter is a substance or an environmental condition, such as chronic inflammation or specific hormonal fluctuations, that forces cells into rapid, relentless division.
So if a promoter acts on completely healthy tissue, the cells just divide quickly but normally.
Right. But if a promoter acts on an initiated cell, it forces that specific damaged cell to clone itself over and over again. It rapidly passes on the broken DNA before the body's repair mechanisms have a chance to fix the original error.
Wow. This completely reframes how we view cancer timelines. Because both an initiator and a promoter are required, a mutation can sleep silently in your tissue for decades.
Yes, decades. You might be exposed to an initiator in your 20s, say, a severe sunburn that mutates a skin cell's DNA.
Right.
But the actual melanoma doesn't develop until a promoting environment triggers that specific cell to start cloning itself in your 50s.
That is wild to think about. And identifying those initiators is basically the foundation of cancer epidemiology. We generally categorize the usual suspects into chemicals, radiation, and viruses.
Right. Chemical initiators include things like aflatoxin, which is a naturally occurring toxin produced by certain fungi on improperly stored grains. It's a massive driver of liver cancer globally.
And asbestos is another potent chemical suspect causing mesothelioma and lung cancer.
Exactly. And then radiation, particularly UV light, directly shatters DNA strands, leading to melanoma.
But viruses and bacteria are also incredibly prolific initiators. HPV is directly linked to the initiation of cervical cancer.
Yes, and a common stomach bacteria, Helicobacter pylori, is responsible for nearly 100% of gastric malt lymphomas.
100%, because H. pylori physically colonizes the stomach lining, causing severe chronic inflammation.
Right, and that localized, endless inflammation acts as a massive promoter. It forces the stomach cells to constantly regenerate to heal the damage, drastically multiplying the chances of a copying error.
So looking at the macro level, the clinical data provides a stark picture of how these agents affect populations. In the U.S., smoking remains the #1 contributory factor responsible for cancer.
By far.
And looking at internal cancers by incidence, how often they are diagnosed, prostate cancer leads in men and breast cancer leads in women.
Yet lung cancer is the leading cause of cancer-related death for both demographics.
Which heavily underscores the lethal efficiency of inhaled chemical initiators.
Absolutely.
We often hear debates about how much of this is strictly genetic destiny versus environmental exposure. There is a deeply profound statistical case study regarding Japanese immigrants to California that really settles this debate.
Yes, it's a fascinating study. Historically, stomach cancer rates were exceptionally high in Japan due to dietary factors, while colon and prostate cancers were remarkably low.
But in California, the exact reverse was true. High colon and prostate cancer, very low stomach cancer.
Right. And when Japanese populations immigrated to California, within just a few generations, their cancer incidence rates completely shifted to match the California averages.
Wow. The high rates of stomach cancer plummeted and the low rates of colon and prostate cancer spiked.
Exactly. The ancestral genetics of the population remained largely the same, but the dramatic shift in diet, environment, and exposure entirely overrode their historical genetic risk profile.
It proves the massive, undeniable role that environment plays in promoting or suppressing cancer.
It really does.
Because environment and lifestyle play such a dominant role, prevention is a tangible clinical strategy. Beyond just lifestyle modifications, pharmacology offers interventions to disrupt the sequence.
Right. Routine aspirin use, for instance, has been shown to decrease the incidence of several gastrointestinal cancers, likely by reducing the chronic inflammation that acts as a promoter.
And there are also more aggressive pharmacological interventions, such as isotretinoin, commonly known as Accutane.
Yes, isotretinoin has demonstrated the ability to decrease the progression of certain precancerous lesions in the lungs and GI tract.
But we have to look at the clinical reality impartially here. The risks are substantial.
Very substantial. Isotretinoin carries severe side effects, including bone softening, depression, liver toxicity, and extreme teratogenic effects.
Meaning it causes severe life-threatening birth defects.
Right. Patients utilizing it must adhere to strict, heavily monitored birth control protocols through a program called IPL-DIG.
So the biochemical interventions exist, but they demand an incredibly careful calculation of risk versus reward.
They do. It's never a simple fix.
Which really highlights just how delicate the balance of our biology truly is. We started today by defining the spectrum of a lump understanding that a confused, misplaced choristoma or an encapsulated lipoma fundamentally differs from a malignant threat.
Right.
We tracked the rogue evolution of a single cell, watching it progress from a reversible hyperplasia into a riotous dysplasia, until it finally manufactured the chemical scissors required to chew through the basement membrane.
We lifted the hood on the genetic engine, seeing how a microscopic typo floors the oncogene gas pedal and cuts the p53 brake line.
And we watched the resulting tumor fight a brutal war for logistics, achieving cellular immortality with telomerase and hijacking the blood supply through angiogenesis.
Understanding this intricate mechanical rulebook fundamentally changes our relationship with the disease.
It really does. It shifts cancer from feeling like a chaotic, random strike of bad luck in the dark into a highly specific, logical, biological sequence.
Exactly. Because it is a rigid sequence requiring both initiators and promoters, it has vulnerabilities. Our daily choices regarding environmental exposures and lifestyle directly intervene in that sequence.
Interrupting the cascade before a dormant mutation is ever promoted into a runaway threat, it replaces fear with an understanding of the mechanics.
Absolutely.
But there is one final, staggering implication hidden inside those mechanics for you to think about. We talked about telomerase, the enzyme that rebuilds the protective caps on our chromosomes. Cancer's ultimate superpower is hijacking telomerase to achieve biological immortality, allowing it to evade the natural death that comes to all normal cells.
Right.
But human aging, the very reason our skin wrinkles, our organs eventually fail, and our bodies break down, is fundamentally driven by our normal cells lacking that exact same telomerase, allowing our chromosomal shoelaces to fray and degrade over decades. If cancer's deadliest weapon is the ability to stop the biological clock, could the very mechanism that makes a tumor so lethal also hold the ultimate decoded secret to human longevity? Something for you to mull over until next time.